I'm really delighted to see you all here for what is the most topical of topics in the sciences right now, the ethics of genome editing, and this event is organised by the Convergence Science Network. My name is Natasha Mitchell from ABC Radio National, where I'm a presenter and a science journalist, currently presenting a new program called Science Friction, which is all about science and culture, so get onto that podcast and download it and tell your friends. You know, the doing of science is not just about scientists, it's about all of us. And when a scientific tool or a technique or a technology lands and changes research agendas overnight, worldwide, that is something that we in the wider public must take notice of. And we must know about it, and we must talk about it, and we must ask scientists about it, because it heralds something pretty big. And CRISPR, genome editing, is one such development. This new technique for editing and modifying and deleting and inserting genes in whatever genome you might consider. Human, animal, plant. Now many of you, who was at the presentation earlier this year by Jennifer Doudna? So a fair few of you, thanks for coming back for this discussion. It was that discussion that has led to this discussion. Such was the interest. And if you haven't seen the video of that presentation, it's on the Convergence Science Network. And of course, Jennifer is the co-inventor of CRISPR from Berkeley and a likely Nobel laureate, I think most people are predicting. And she has come to be very interested, very engaged in the ethics of this technology that she's responsible for in part. So we have CRISPR allowing scientists to more effectively study, investigate, understand genes that work in our bodies, other animals' bodies, in plants. It's allowing scientists, and many of you will be scientists in the room. Who's a scientist? Great. Who's using CRISPR? Soon. Excellent. It's allowing some of you to study early development. It might also be useful as a therapeutic tool one day, as you know, allowing disease-causing genes to be tweaked or deleted, and one day down the track, and this is purely speculative at the moment, it might also allow us to modify the genetic makeup of human embryos before they grow to full term. So as a reproductive tool. And CRISPR leads the way in some very interesting developments. It really points the way to some interesting developments, but as we've all considered, it raises some very interesting ethical questions. Who will have access to this technology? How will it be used? Who should use it? Who should get to say how it's used? How should it be regulated? How should it be monitored, controlled? What's the social impact? So tonight we're going to hear a 30-minute presentation from a bioethicist, and then we're going to open out the discussion to involve Louise Sales, who's a scientist, a biologist, and she's the coordinator of the Friends of the Earth Emerging Tech Project, and also Melbourne University geneticist and researcher Professor Phil Batterham. And then we're going to open it out to you for questions and conversation as well. So to introduce our speaker that will provoke and get us going, Christopher Gyngell is a research fellow in biomedical ethics at Murdoch Children's Research Institute and at the University of Melbourne. He was previously a fellow at the University of Oxford, where he undertook a fellowship focused on this very topic, the ethics of gene editing. He was awarded his PhD in philosophy from the ANU in 2015, and before doing his PhD, he completed a master's degree in applied ethics and an honours degree in genetics. So his pathway has been very interesting to get to this point. Please welcome Dr. Christopher Gyngell.
Great. Thank you, Natasha. I want to first of all thank the Convergence Science Network and Luan for inviting me to speak today. This is obviously a topic I'm really interested and passionate about, and it's amazing to see such broad interest in the public, and it's great to get these public discussions going in this interest of this topic, because I think that's really vital going forward. For decades, scientists have been able to alter the genetic makeup of many species of animals. When I was young, I'd often see images like this on the middle there of the glow-in-the-dark mouse that was created by splicing the genes of a jellyfish with the genes of a mouse so it glowed in the dark. Perhaps the most famous transgenic animal that they created with these early techniques was the super mouse, who is at the top there. Now, the super mouse was created by modifying a receptor that's to do with metabolism. Essentially, that mouse could run for 6 kilometers, which was much further than the 200-meter limit of a normal mouse. Beyond that, they could live much longer than a normal mouse. And in addition, they were more fertile. They ate twice as much as a normal mouse, but they only had half the amount of fat. So it was quite good to be a super mouse when you got it. In addition to altering the physical characteristics of animals, they also were able to alter the social characteristics of animals with these characteristics. Down the bottom there is a pair of voles. They were a normally polygamous species. They had numerous sexual partners. But again, by altering some of the genes, they could make this polygamous vole a monogamous vole. So you can see there this nice monogamous couple of voles cuddling, forming their lifelong pair. So these techniques were great for science, but they had serious limitations. They were very inefficient, so you would only get the change you wanted in a small number of cases. You had to use a large number of animals to get the precise change that you wanted, and they were also very imprecise. They would make a large number of changes to the genome in addition to the one that you wanted, and for those reasons, they had really limited potential to be a useful modifier of human DNA. But in the last 6 years, we've seen a real revolution in genetic engineering science. A variety of techniques have been developed that allow you to make precise and efficient changes to DNA, and they have been given the collective moniker genome editing. The most famous, the most well-known genome editing technique is the CRISPR-Cas9 system. This consists of 2 components, the CRISPR molecule, which is like a guide molecule that can bind to different DNA sequences, and the Cas9 molecule, which is like a pair of molecular scissors. This system is found naturally in bacteria, and it's used as a defense mechanism against invading viruses. What Jennifer Doudna discovered in her lab was that you could alter this sequence so it could target virtually any DNA sequence in a cell, and then furthermore, once you had made the cut to the DNA, you could recruit the cell's own DNA repair mechanisms to add, take away, or to alter the DNA sequence there. And it has been a really remarkable technology with a wide range of applications. So I'm here to talk about the ethical aspects of these applications. And I think it's really interesting to go through the different types of things genome editing can be used for, because they all raise different sets of issues. And some of the things it's used for raise quite familiar ethical issues that I think we understand quite well. And in particular, its use in somatic medicine. So to make changes to our DNA for medical purposes in a ways that we won't pass those changes on to our descendants. So there's numerous trials underway worldwide, most of them in China, which use this technology to try and cure cancer cells. There's been some really exciting studies in leukemia where it seems to have been able to reverse leukemia that was at a very serious stage. And this is very exciting, but it also raises very challenges. So this is a picture of Hosea Zayner, who claims to be the first person to ever have injected himself with CRISPR. So Hosea tried to inject himself with a CRISPR that he designed to knock out the enzyme myotosin, which normally inhibits muscle growth. So you'll see the dog there, that's a CRISPR dog that's had myotosin knocked out. And you can see it's gone from a nice, normal little skinny whippet to quite a beastie big whippet. And he is trying to do that to himself. Now, this is obviously very iffy. This is a big ethical problem, and I don't recommend it. But we understand this problem well. It is bad to inject yourself with untested medical products. We need to have regulation, we need to have safety trials, we need to have clinical trials. But CRISPR, in addition to these familiar problems, ethical issues, it raises quite novel and quite more profound ethical issues. And 2 areas where this has been sort of identified are its use to alter germline cells, so embryonic cells, stem cells, egg cells, basically DNA that we can potentially pass to our descendants, and also its use to alter animal species. And that's what I'll talk a bit more about in this presentation. I'm going to use the term of the acronym GGE to stand for germline gene editing. Now, what I mean by that is you're altering germ cells, and this can be done either in a purely research capacity, altering embryos in a petri dish, or in a reproductive capacity. And I'll run those things together because often regulations and laws actually combine these two uses. So in 2015, a lab in China became the first to use CRISPR to alter germline genes. They use it to alter tripronuclear embryos, which have 3 copies of a chromosome in every single cell. So they're unviable embryos that would normally be spontaneously aborted early in pregnancy. And they tried to use the CRISPR molecule to reverse the mutation that caused beta thalassemia with mixed success, but it was a hugely controversial study. The leading science journals, Nature and Science, both ran opinion pieces arguing that this research was unethical and should be stopped. There should be a moratorium on such research. Jennifer Doudna herself was a co-author on one of those papers. But from that, other studies have happened. I've got here 5 studies have now occurred with CRISPR-Cas9. I did read on Twitter, on the tram here, about a 6th one, which happened about a month ago. In addition, there's been other uses of other gene editing systems that aren't CRISPR-Cas9, but are based on the CRISPR molecule. So research is continuing. Why would you want to do germline gene editing research? Well, it can be really important to ask some vital questions about development. Kathy Nykown, who is at the top there, she's from the Francis Crick Institute in the UK. She used this technique to really study early development. So we don't really understand the process where a human goes from a single cell pluripotent stem cell to this complex multicellular organism. And by using this gene editing technique in these studies, we can turn on and off particular genes and get an idea for the mechanisms that control this. This is interesting for its own self, but it's also could lead to treatments for infertility, to better IVF success rates, and also give us a better idea about the high rate of spontaneous natural abortion loss that occurs in humans, and that's precisely their motivation for conducting this study. But despite this, this is quite widely sort of banned process. Here's an image from a recent article that looked at gene editing laws in around 39 countries, and 75% of them had bans on germline gene editing, and most of those apply to research purposes as well as to reproductive purposes. But when you get to the concerns about research, a lot of people will go away from the research and they'll say, no, well, what we're really concerned about is the reproductive applications. If you look just at the research, though, most times when I've spoken to people about it or had debates, the only really thing that comes up is the use of embryos. Obviously, this research does need to use embryos. If you're of the philosophical belief that a human embryo has a full moral status, it's like doing a research on a child, then of course you're going to find problems with these early studies. But it's important to note that this concern isn't unique to germline gene editing. The NHMRC has currently about 8 active licenses for germline gene editing, for research on embryos. And germline gene editing would sort of do this research just with a different technique, would be the only type of thing that would be different about it, but yet it remains banned. Furthermore, as I've sort of just touched on, the use of this technique might also save other embryos or reduce the use of other embryos in other techniques. So IVF and PGD, you automatically lose embryos. And also, some of this research is sort of designed to understand sort of natural embryo loss in pregnancy. So I don't think there are particularly good reasons about sort of banning this research. Again, we're talking about research that occurs purely in petri dishes that will never be implanted into women. Further on down the track, we might want to use germline gene editing for disease prevention. the most immediate use for this would be to do reverse changes in monogenic diseases, so diseases that involve a mutation in a single embryo, in a single gene. The World Health Organization estimates that over 10,000 diseases are caused by defects in a single gene, and potentially, if germline gene editing is proved to be safe, we could use it to correct these mutations very early in the embryonic life. Of course, we need to wait until this technique is proven safe, but it's a very early technique. As I said, it was only sort of developed 6 years ago, so it stands to reason that as more research is conducted and as we get more and more familiar with it, could be shown to be safe for this purpose. We already have a technique that can avoid genetic diseases, monogenic diseases. This is pre-implantation genetic diagnosis. So how that works is you create a range of embryos through IVF, test each one for the presence or absence of certain diseases, and then implant the ones that have a desirable genetic makeup. But unfortunately, PGD isn't always effective. So in around 17% of IVF cycles, only one viable embryo is produced. And in those cases, selection is not possible. Furthermore, some people are homozygous for a dominant condition like Huntington's disease. And in that case, all of the embryos they produce through IVF will carry a disease-causing mutation. So again, PGD wouldn't be an effective treatment for them. People often object when I mention these points that such cases are really, really rare, and it's true, it's completely true, they are rare. But something being rare doesn't change the moral valence of preventing it. Just because you have a really rare disease doesn't mean you don't have moral reasons to cure that disease. And same here, just because this is rare doesn't mean that we should potentially deny people a treatment that can allow them to have children free of genetic disease. So I think this will be the first type of reproductive use of germline gene editing, who knows when, maybe 5, 10 years down the track. But it will probably be used as a package with PGD. So if PGD isn't an effective treatment, you've only got one embryo, then you can use gene editing to correct. In the longer term, and I mean probably several decades from now, GGE could be used to maybe target more widespread causes of death around the world. So 30% of all deaths in those under 70 worldwide are caused by chronic diseases, such as heart disease, cancer, and diabetes. Now, I think as an ethicist, that's a really bad thing. We all know some people that are innately resistant to the perils of aging, that can live successfully into their 80s and 90s, and I think the world would be a better place if more people were like that. So I think we've got good reasons to pursue these sort of quite speculative avenues to preventing premature death, and I think gene editing research into this will be one of those avenues that we should pursue. Some people have concerns about using germline gene editing in this way. I won't be able to go through all of them here. But in general, a lot of people try and endorse some sort of principle of non-interference, that we shouldn't alter an embryo in such a way that's going to make a big difference in its life, mainly because we can't get consent from that embryo to do that change. And these principles seem like really good ethical principles when we're talking about two consenting adults. I shouldn't just be able to go up to someone or change their hair color or change their eye color without asking them first. We might even say I shouldn't just cure them of a disease if I had that power without asking them first. But it seems to me a really bizarre principle to apply in the context of a parent-child relationship. I mean, parenting is all about interference. Parenting is all about making sure your child grows up to be healthy and happy. Some people will come back at this and say, well, we should draw a distinction between environmental influences on a child and genetic influences on a child. But I think when you really look at it closely, these distinctions get really, really tricky. And I'll give you one example, which is taking vitamin D. So many health systems around the world and in Australia recommend pregnant people take vitamin D. The motivation for this is to prove the health of the child. So it protects against bone diseases like rickets, and it might also protect against autoimmune diseases. But we also know vitamin D is an epigenetic modifier. It can change gene expression. And there is some evidence to suggest that it can change gene expression in the developing embryo. So if you have this view that we shouldn't interfere with a child even to prevent disease, then you come up against these hard other cases where it looks like we really do have good reason for women to take vitamin D supplements, and we do have good reason to alter embryos in such a way that it promotes their health. By a large margin, the most controversial aspect of germline gene editing is human enhancement, going outside of disease, treating disease. From super mouse, we'll have superhuman. And I should tell you now at this point that when I talked about the super mouse earlier, super mouse had some other characteristics which I left out, one of which they were incredibly aggressive. They would attack other mice very unprovoked. So it shows you that once we start to leave using this for treating disease, we start to take aboard more and more risks, I think. But people do argue that we will have strong reasons to do this. One of the reasons we have moral reasons to prevent disease is because we have reasons to promote the well-being and welfare of our children and promote their happiness. But some of those things will be achieved through types of enhancement by maybe giving them better memory, better vision, making them stronger or taller. So we have that type of motivation to improve the well-being of individuals through enhancement. Another range of sort of arguments say, well, not so much for individuals, but we'll need this tool at some point in our species' history. You might just think if the worst predictions about climate change are true when we get into a very, very different environment, perhaps the atmosphere starts to change. You know, to keep surviving and thrive, we might need to make radical changes to our bodies. So some bioethicists have argued that we might even need to radically modify ourselves so we can travel through space and colonize other planets. I know I'm getting very sci-fi here, but just letting you know what's sort of out there. Again, these are some of the reasons that we think we might want to engage in enhancement, but there are lots and lots of concerns about this. I think some are better than others. A range of concerns come up about we're going to disrupt parental relationships through these technologies, through so-called designer babies. So rather than just picking the sort of accessories for your nursery, you'll soon be able to start to pick the traits of your child, their eye color, their hair color. And this is meant to be bad for the child because they start to be treated like a means rather than an end in themselves. But it's also bad for the parents because it might deny them from developing important virtues such as openness to the unbidden. Another range of concerns about enhancement focus on the sort of justice, the social justice angle. Right now we live in a world that's marked by inequalities, but these inequalities are generally social in nature. It's access to money, access to goods and services. But what enhancement through germline gene editing threatens to do is to write these social divisions into our very biology. So rather than just being divisions of access, there'll be divisions of ability. I'm not sure. I think there are definitely ways you can sort of mitigate these examples. Inequality is generally, it's a political problem. It's a political problem of equality of access to a good, and there are political solutions to it. You make sure the good is accessible widely. Can be harder said than done, but again, it's more of, I think, a political problem than a strictly ethical problem. The last sort of concern I've got there is just I bundle them as externalities. I think once you start talking about enhancing things rather than treating disease, which we know we've got strong moral reasons to do, all these sort of other little risks, I think, start to pile up. One of these, I think, is collective action problems. So I'll give you an example of height. Say you could use gene editing technologies to make your child taller. There's good reason to do that. A lot of studies show that subjective well-being is boosted by height, particularly in males. So parents might all want to make their child a bit taller. But of course, the problem is if everyone does that, then everyone gets a bit taller. Your child doesn't get an advantage. But everyone's just a bit taller, and no one gets an advantage. But this could keep going for the next generation till we're very tall and ungainly, and we've got to redesign everything, and it's really, really bad. Another type of thing, I think, is in cognitive traits. So lots of studies have now been done in social resources that show that cognitive diversity is more important for groups to solve complex problems than individual ability. Now, once you've got lots of people choosing cognitive traits for their children, you might get this situation where we're all choosing very similar cognitive traits, and we're making ourselves individually smarter, but collectively dumber. And I think these things really need to be sort of considered. Most of my research, I should say, has been on germline gene editing, on CRISPR in humans, but I know a lot of my fellow panel members are more experts in using it in animals, so I'm going to just briefly run through some of the uses and some of the concerns. So there's been a number of potential applications for gene editing in animals. Some of them have been in agriculture. I think some of them sound like really good ideas, particularly making animals that are naturally resistant to diseases so that we can use less antibiotics. Antibiotic resistance is a huge global problem that doesn't get reported nearly as much as it should, but it does threaten to kill millions of people going forward. One of the largest contributors, I think it is the largest contributor to antibiotic resistance, is its use in agriculture, of farmers just regularly giving their stock antibiotics to protect them. Potentially, gene editing could help solve that problem by making animals more resistant, especially in livestock, more resistant to diseases. Another potential use is to reduce animal suffering in agriculture. One of the first sort of CRISPRed animals was a hornless cow, which you can see there. Now this is designed to promote welfare, because when cows have horns and they're all done in a paddock together, they tend to gorge each other, which is obviously very horrible and bad for them. That's a very slight, easy fix, but as you go more sci-fi, you can think, well, perhaps we could create animals that just didn't suffer in captivity in livestock. Perhaps they even would enjoy being kept on a farm. I think they're interesting things to think about. Food security, so again, another early sort of CRISPRed agriculture item was drought-resistant wheat that could grow even in drought conditions. And again, I think if it's promoting sort of food and food security, then it seems like they've got a good idea. They've also been used in conservation biology, well, they've also been suggested to be used in conservation biology. I think this is a lot more risky. I think with agriculture, with a lot of those animals, we've already altered them so much, and they all sort of live with us. It's probably okay to alter them a bit more. Conservation biology, you get bigger risks. One of the proposed uses has been to do in predator control. So New Zealand have this really sort of ambitious program of a goal of becoming predator-free by 2050, so wiping out all the mammalian sort of predators like possums, mice, and things. It was proposed that they could use gene editing to do this through a system called a gene drive. Now, a gene drive is a system where you use CRISPR, you make a change, and it certainly guarantees that that mutation is then passed on to future generations. This was originally thought, well, maybe we could use this to sort of put into a natural predator population, like a possum population, and then it will build up. One of the people that suggested this then wrote another paper and said, actually, I've thought about it more, and this is a terrible idea. So we'll figure through how it goes. So say you do it for possums, which are a predator in New Zealand. You introduce this gene, it spreads, and now they're all infertile and they can't breed. But what if that possum waned its way to Australia? Then all the Australian possums would go, and so on and so forth. So I think these things, when you're talking about altering in this type of thing, raises a lot more risks. So that's basically it. Look, so genome editing is a powerful technology which presents us with enormous opportunities and challenge. My main view is that we should really look at each application on its own merits and avoid sort of sweeping statements that it's good or it's bad. We need to look at each application on the basis of it is. And I think for some applications, we will have good reasons to use it. That's why I'm against sort of sweeping sort of bans, particularly bans on research that are in place in many parts of the world. And yeah, I think right now we've got great users to use it in research, and potentially in the short to medium term in reproduction to cure disease. And thank you, that's my view. Thanks.
Thank you so much, Christopher, for that overview and presentation and sharing some of your, I guess, interpretations of the ethical landscape that we're heading into. So now we want to open out the conversation to invite 2 other people into the fray, and then I'll open it out to you. We've got some microphones that will get you involved in about 25 to 30 minutes from now. First of all, we have Louise Sales with us, who's joining us from Hobart. Louise has coordinated Friends of the Earth's emerging tech project for the last 6 years, and the focus of their campaigns are on calling for a precautionary approach and an equitable approach to new technologies like genome editing and synthetic biology. Louise has an honours degree in biology and a master's degree in biodiversity and conservation, and she's has over a decade's experience campaigning on tech issues with NGOs. And Friends of the Earth have written some rather interesting documents, which I encourage you to go and look at. We also have Professor Phil Batterham. Professor Batterham, Phil, is from Melbourne University and from the Bio21 Institute. He's a global research leader in the genetics of insecticide resistance. Think of those mosquitoes and any other pest. Lots of them. And he's current president of the International Genetics Federation. He is a 5-time finalist in the Eureka Prize for Public Communication of Science, and he's organised a host of conferences, including some years back the International Congress of Genetics and the UN Conference on Global Health. So please welcome our next panellists. I might just whip through you quickly with, I'd like you to say one thing you're hopeful for in relation to this technology, and one thing you're concerned about. It doesn't have to be everything, that's a hard one, I know. Just one thing you're hopeful for and one thing you're concerned about, but we'll unpack more of the issues as we go. Phil.
Something that's not been clear in the discussion yet is that CRISPR is an incredibly powerful research tool in fundamental research. And through that, it could have amazing impacts on human health without ever going near a human. So that's one thing I would say. In terms of concerns, I would endorse a statement made on the last slide that you need to consider applications on a case-by-case basis, and you need to consider all options as to what you might do, including gene editing, but other technologies, other options for managing an issue.
Louise.
Yeah, I'd say I'd share the enthusiasm with some of the potential medical applications of gene editing, but I would be deeply concerned about human germline editing and also gene drives, as you've mentioned, and also some of the other environmental applications where you're actually really seeing genetically modified organisms into the environment. That would be where my particular concerns would lie.
Chris, you've had a go, but just briefly.
I'm very pro for it to treat disease and to avoid genetic disease, but within that, I'm also concerned about its use for that purpose in an unregulated way. So I think the scariest thing for me right now is in this technology in its young stage where it's not ready to be used reproductively, you get in rogue sort of fertility clinics set up in jurisdictions where there's not good oversight, and then offering to sell that to patients when it's not ready and it being unsafely used like that.
They're offering to sell it, or they are selling it?
They may offer to sell it. That's what scares me, or selling it, yeah.
And it's potentially coming, isn't it?
Very, yeah.
So we'll unravel some of those concerns that you have and the optimism you have for the technology as well. Phil, just take us inside your lab to give us a sense of how CRISPR has changed your research agenda effectively overnight, as is the case for labs worldwide at the moment. So what are you up to?
So I work on a so-called model organism. So this is a vinegar fly. You might see it on fruit at the market. You might see it around the rubbish bin. It lives wherever humans live on planet Earth. And when Gregor Mendel's principles of inheritance were rediscovered at the start of the 20th century, people used to look for mutations in genes, but they had no way of making them at all. So they would just wait and they'd look at flies and they'd find spontaneous mutants, and they'd find a fly that had, for example, white eyes rather than red. And using that mutation, that particular one, they in fact discovered that genes were on chromosomes, and there was such a thing as such a thing as an X chromosome. And in fact, that led to a Nobel Prize in medicine.
So we like our flies.
Yep. Now, they just had to sit around and wait. And then in the 1930s and 40s, there was a man named Hermann Muller, and he discovered using this fly that you could use X-rays to create mutations. But they were random, and the spontaneous mutations were random. And then people started to develop some tools in this organism where you could target mutations to particular genes, but they had the downsides that we've heard about. And then comes along CRISPR, when we can create, and every student in my lab is doing it, creating mutations of any type in any gene that we want. So we look at insecticides and we look at the mechanisms by which they work. So we find the receptors in the nervous system that are being targeted by some of these insecticides that concern people. For example, the neonicotinoids. And one of my students is sitting over there, and he's focused very much on the targets of this drug.
So one of the most common insecticides. In fact, 3 of them have just been banned by the European Union.
And biopesticides, I should say, that do the same thing. Organic insecticides that do pretty much the same thing and are even more toxic. But we're also able to use this technology to look at downstream effects that might impact beneficial organisms that are impacted by the insecticides as collateral damage.
So honeybees?
Yes.
Wild bees?
Yeah, and not just honeybees. We've got a situation where there's population collapse in a very large number of insect species around the world, and some of them are known to be beneficial, and many of them we just don't know what good they might do in the environment. So it's very much a research tool. Now, in the same organism, there have been several Nobel Prizes in medicine awarded, and in that very same organism, people have, in the last few years, discovered countless genes that impact human health, that are responsible for disease in humans.
So what has CRISPR, this new form of gene editing, particularly allowed you to do that you couldn't do the day before?
Just to create a customised mutation. But
how accurate is it? And how easy is it to do that?
Well, people talk about off-targets because there is this guide RNA that has been described already, and it takes basically the cutting enzyme Cas9 down to the DNA. Now, the guide RNA sequence isn't very long, so people have argued that there could be many other places in the genome that would be cut.
Well, they haven't just argued. I mean, people find off-target mutations.
They do.
In other words, you say, well, I want to go in and I want to edit out that particular gene. Oh, hang on, that gene over there's been edited out.
Yep.
Oh, hang on, that gene over there's been edited out.
Well,
so that's still a concern, isn't it?
That is a concern, and if you were working on a human, you'd be very concerned about that. We're not so concerned in a fly, i.e. there's not the ethical concern, but it does mess up our interpretation of our data if we've got multiple mutations in the genome. So we do check those off targets, and I'd have to say that we don't find them. You know, we find that in our hands, in our system, this technology is very precise in terms of where the cutting is occurring.
I guess there's another issue, independent of all the other ethical issues that we've talked about in relation to human germline editing, which we'll come to. You are creating in your lab organisms that are insecticide resistant. And so there's a biosafety question there.
Absolutely.
You need to know what you're making quite precisely and contain it.
And that's exactly why I don't use a pest. So it would be, so in some places people do make insecticide resistant pests. I think that's totally unethical. And they've done that without CRISPR.
Because if they get out into the world, you don't know what they're going to do.
We have mutants that literally would be as impacted by water as insecticides. They're so resistant. but we're not working with a pest, and we do have, there are very strict containment procedures even for these organisms that are not a pest. So we have protocols for managing these organisms for which we've edited the genome.
So, Louise, one of the things that you're focused on with the Emerging Tech Project at Friends of the Earth is the regulation question. I guess the containment question, in a way. Regulation, how this stuff gets regulated, is a massively hot topic across the world right now. And there's a lot of uncertainty, and most people's legislation around the regulation of genetic technologies is under review. In some countries, they've made a clear decision. We haven't yet. So you're keeping a very close eye on how these technologies get regulated, aren't you?
That's right. So as you say, currently these techniques are regulated, but our gene technology regulator here, the Office of the Gene Technology Regulator, actually released a draft proposal earlier this year to actually deregulate these techniques like CRISPR when it's used to make small additions or deletions to the genome. So that would mean that anyone in Australia could genetically modify animals, plants, microbes with absolutely no regulation at all. So that's obviously something that we're really deeply concerned about, particularly as the Office of the Gene Technology Regulator acknowledged that even really small changes to microbes can result in huge increases in pathogenicity. So that's a major biosecurity concern. There's obviously ethical concerns as well if anyone's allowed to genetically modify animals. It would make Australia the first country in the world to deregulate genetically modified animals. And plants as well. So there's, if one of the major concerns...
There's a salmon that has been
approved.
It's not on the market in the US, but it's been approved.
Yeah, that's true.
A genetically modified salmon.
But these would be entering the food chain with no labelling and no safety testing, so we're actually deregulating. That salmon is regulated.
So let's unpack that a bit more, because the question of regulation is there's lots of subtleties involved to this. So at the moment, genetically modified organisms are defined as GMOs if they've been created using a genetic technology.
That's right.
A gene technology. So that's where it's at. So what are they actually proposing to change?
So they're proposing that if techniques like CRISPR are used to make what they say are small additions, small deletions to the genome, that they shouldn't be regulated. They're saying that these changes could occur through mutagenesis, which isn't regulated, or natural breeding. So they look the same, so therefore they are the same. But as you mentioned, all of these techniques have off-target effects. They all involve in vitro DNA techniques, so they involve a petri dish. They involve the use of all the GM techniques, so particle bombardment, and all of these can result in unintended mutations. In the case of plants, this could lead to the production of novel toxins, novel allergens. So we find it, frankly, astonishing that the government's actually proposing completely deregulating these techniques, and it's completely at odds with what other governments overseas have recommended.
So let's unpack that even further, because I don't think there's a proposal to totally deregulate, but it's to differentiate between, are you editing an organism in a way that just is involved with taking out an existing gene or adding a gene from an equivalent species, or are you using it to add a gene from another species altogether, which is a transgenic modification? And that's, so there's all these distinctions. So Phil, what particularly are scientists calling for in relation to regulation?
Well, I don't think there's a standard position that we've all signed up to. My work is currently in the CRISPR area is regulated like any genetic manipulation. So, I have a license to do what we do through the Office of the Gene Technology Regulator, even though, very few people would have any concerns about what I'm doing. But it is still highly regulated. I'm quite comfortable with that, and to some extent, I want to see the public comfortable with what we're doing, and there are perceptions and there are realities.
But what about some of the concerns that Louise has about the potential deregulation of CRISPR? So, in other words, CRISPR would be free of regulation unlike other genetic technologies before it?
I don't believe that's going to happen. I think it's more subtle, as you've indicated. There are different levels of intervention that will be regulated differently. But as a scientist, I'm very happy to live within the frameworks that the community places upon me. And scientists should be in that position, because there needs to be really community support for what we do as researchers. So I do not have any concerns about regulation. It imposes extra work on us, but if that's what's required to make the community feel comfortable, then I'm happy to go there.
Well, we'll come back to the question of whether the community's been involved in the consultation around the regulation. Chris, what's your take on regulation as an ethicist?
I like regulation. I think it's
You wear your
very
seatbelt.
good. Yeah. I think it's, a lot of when I find I have different views of other people around the world, it comes down to, I think, your trust in government, and I've got a really good trust in government. I think governments can make good decisions and can control these things and make
But in
sure
relation
that
to CRISPR in particular,
yeah.
have you been tracking the discussion around regulation and what should and shouldn't be controlled?
Yeah, like I would like a licensing system, I guess, where you propose a use, it goes to an expert panel, they review it, and they look at the risks and look at the benefits and make an informed decision. I think that's quite a good use. I thought it was, I know in the US, they had, I thought they had like wheat crops actually hit the market that were CRISPR'd because they didn't fit they didn't classify as genetically modified organism because there was that loop in the...
There's a canola crop, it's not a CRISPR one.
Right, okay.
Yeah, and you're right, because the regulatory regime in the US, under the USDA, it has to be deemed a plant pest to be regulated as a GMO, so they've got slightly different regulatory system to us. But I think
even though it was edited, I think it didn't fit that regulation because their regulation said it had to have DNA from 2 different species together. And that obviously is quite silly. If you've got something for genetic modified organisms, you should include ones where you're going in and editing the...
Yeah, I mean, this sense that the argument, I'll get Phil in on this too, if the argument being made in terms of regulation is that that not all products of CRISPR are necessarily worthy of the label GMO.
Correct.
So you don't regulate the process, you regulate the product, is the argument being made.
I could give you an example of that.
But if you apply CRISPR to any organism, you are modifying its genome. Is it not called a genetically modified organism then?
I could give you an example of the equivalence. So the first time that my lab published a paper on it, we found a mutant that was resistant to an insecticide, and this resistance was dominant. This was a bit unusual because it meant that only one copy of the gene had to be have a particular DNA sequence that would confer this resistance to insecticide. You didn't have to have 2 copies where normally you would. So we thought, we sequenced the gene and we found this mutation, and we thought that it was causing, this particular mutation was causing that dominant resistance. But to prove that, we had to recreate that, and we used CRISPR. So here was a mutation that we'd isolated by other means, and we went back and reproduced that mutation in a different way using CRISPR. So the first mutation that we generated would not have been regulated in any way at all. The second one was regulated because it was a targeted genetic intervention. Now, I still don't have problems with that. Where I do have problems with, regulation in the lab doesn't concern me much at all. The regulation of things that happen in the field, I think in the area of genetic modification, in agricultural crops, at some points the level of regulation has been literally insane. And I think the level...
What do you mean?
Oh, well, in some parts of the world, and just for example, in India, there was a case of genetic modification that involved putting in resistance to insecticide into cauliflower and cabbage crops. And part of the regulatory requirement is you would feed cattle, which are sacred animals, massive amounts of cauliflower to test whether it was safe. So much cauliflower that the animal would almost die if you tried to feed it. There are hoops that you have to jump through with some GM crops that I think...
Hoops or collecting best evidence?
Repetitively collecting, in some cases, the same evidence about safety, for example.
Louise, so the question of who gets to have a say in the regulatory regime in Australia and overseas is an interesting point. And you at Friends of the Earth have been submitting a whole stack of freedom of information requests. Why?
We were concerned that this whole discussion so far has been going on behind closed doors. There's been expert panels advising the government on how these techniques should be regulated. So we actually got a...
It's not entirely behind closed doors. There's been a public submission process for the major reviews.
Yeah, prior to that, there was a Gene Technology Technical Advisory Committee, which basically recommended to the Office of the Gene Technology Regulator that these techniques be deregulated. So we got freedom of information documents and found out that two of the panelists on that expert committee had basically recommended deregulating these techniques, and both of them had serious conflicts of interest. So one of them was in a strategic partnership with Monsanto. They had patents in these techniques. Now, under the Gene Technology Regulators' regulations, members of their expert panels are actually required to, if they've got a conflict of interest, to not take part in decision making, but they clearly hadn't followed this process yet. Those recommendations basically informed the OGTR's entire discussion paper, the way the whole debate was framed, and their subsequent recommendations to deregulate these techniques. So we're concerned that that's a serious breach of process, that you're basically letting conflicted scientists write the rules on GM.
How do you define what is a conflicted scientist in this conversation? So if you have an advisory panel advising the review of gene technologies like CRISPR, you need people with expertise who understand the technology to be part of that conversation, don't you? They're there as an expert advisory panel.
You do, but these people had personal patents in crops. So if you're wanting to commercialise a GM crop, you don't want it to be labelled as a GM crop, then, because that's what the main resistance to GMOs has. In Australia, the majority of Australians simply don't want to eat GMOs. And industry has decided that, these new GM techniques such as CRISPR is actually a really ingenious way around understandable community resistance to GM crops. We'll pretend they're not GM at all. They won't be regulated, they won't be labelled. Consumers will have no way of avoiding them. And that's industry's plan, has been industry's plan all along with these new GM techniques. So if you look at the language that they use, it's all about these techniques are precise. But in actual fact, the science shows that's not true. There's off-target effects associated with all these techniques, which is why it's really, really important that they be regulated, that we look for these unexpected effects. One potential unexpected effect that I discovered just the other day when I was looking through the literature was there's a concern that you could actually accidentally produce a gene drive in an organism. So if the CRISPR gene gets incorporated into the genome, then it can basically replicate itself in all the progeny. And in
a wild population, which is why it's useful for predator control,
which is why it needs to be regulated. Yeah, I mean, it's really important that we establish that there hasn't been any accidental integration of CRISPR cassettes into the genome, that there haven't been in the production of novel toxins, novel allergens, that there aren't potential animal welfare implications for animals if we're going to go down that path as well. So
just one thing about off-target effects that needs to be stated clearly is that if it's in an organism that you're producing, say, for release in agriculture, you will discover it before it's released. You'll sequence the genome and you'll be able to show that either there is or is not an off-target effect.
Only if you're required to by regulation. There's no, if you're wanting to just rapidly commercialise these traits, why would you bother sequencing
well,
the entire
if you go back to regulations around GMOs, you had to characterize the site of insertion of the element, and you had to show using gene expression profiles that there were no other changes in the genome. But now that genome sequencing is so inexpensive, I'd be pretty certain that you would have to show that that change was the only change.
No, well, you'll only have to show it if it's regulated. Is that the case?
That's right. Yeah.
So if it's not regulated, it would be required through regulation, but it would not be a particularly onerous regulation given the cost of genome sequencing. Yeah. So if you wanted to impose that as something that had to be done under regulation, I don't think you'd even get a whimper of resistance, simply because it would be so easy to do.
Well, the biotechnology industry's been lobbying really strongly for these techniques to be deregulated. They've been saying there's a regulatory burden that they can't cope with, despite there being a huge explosion in CRISPR research in Australia.
Okay, let's get a response from Christopher Gyngell.
Well, I thought it was just to play sort of devil's advocate, because I guess some of the risks you're talking about, we don't know if there's going to be new viral kind of interactions, we don't know how it's going to react, but that's surely true of any novel crop that we release through traditional methods. So is the argument that, well, I can just breed a new crop of cannoli and I expose it to mutagens, I find the funniest looking ones, whatever, and I do it, and in that case the end up product has all these random mutations in it. So is the concern that we should be putting those breeding ones through the same regulationary steps as ones that we use the technology to get to the same?
Well, I think with traditional breeding, you're just working with the existing genes that you've got, and the actual natural mutations actually is incredibly low. I think they reckon that a chance of one gene in a crop mutating in a generation was something like 1 in 400.
But genes are mutating million. All the time in organisms.
But not as quickly, and you're not driving them quickly to commercialization. So I guess the real power with these techniques is you could actually sequentially engineer an entire genome to produce the anthrax toxin if you wanted. You edit one gene, go back, edit another, and if you do that sequentially, you can make really quite dramatic changes to the genome, but at no point in that process would that genetic engineering be regulated. And that's the concern here.
I mean, speaking of anthrax, you've also been interested in the defence funding for some of the research that's being done, keeping in mind, I guess, that defence agencies like DARPA in the US, they fund an awful lot of different aspects of research, from neuroscience to genetic engineering. But what are your concerns there?
Yeah, so the research that you're referring to is actually some gene drive research that CSRO and the University of Adelaide are doing. They're wanting to drive populations of mice to extinction on offshore islands in Western Australia. And very few people actually know about this, and they're actually getting funded by DARPA, which is basically the research arm of the US government defense agency, to do this research. Now, DARPA wouldn't be funding this unless there was a military application, and the military application of gene drives is you could...
Well, a potential military application.
Chris? I think DARPA fund ethics research too. I just, back on Natasha's point, I think they do fund a vast array. I just remember being surprised I go to an ethics convention and someone's funded by
But there are definitely
DARPA.
dual-use applications of gene drives. I mean, you could drive local populations to extinction. You could drive crops to extinction, release insect pests. I mean, there's
well, and the founder of the developer of gene drives, Kevin Esfelt, who was out here in Australia with Jennifer Doudna, he's acknowledged he's got great concerns about the possibilities of that technology, at least. Chris,
Natasha,
did you have something to pick up on? I'm just mindful that, Chris, you were going to say something earlier.
Oh, no, that was fine. I mean, I think with gene drives, I do find the potential applications different. So I think it does seem just inherently risky to me if you've got a gene drive and trying to wipe out a species. That type of application does seem much riskier than trying to reduce antibiotic use or trying to make a crop.
Kevin Esfelt's actually, who's the designer of gene drives. I mean, he basically invented them. He's actually conceded himself that they're too risky to be released into the environment. Yet we've got the CSRO actively planning to release gene-derived mice into the environment in Western Australia. One of the islands that they identified for release, which again we found out through freedom of information laws, is it's 1 kilometre from the mainland. I mean, people go there on their kayaks, and I mean, the idea that somehow those mice would be contained on that island is just preposterous.
Well, I mean,
It's
Kevin Esfelt's
really
interested in having community dialogue around communities within which the technology might be deployed. Phil Batterham, what were you going to add?
Well, in any problem that you've got, whether it's these mice, whether it's insect pests or whatever, I think you have to be really objective and line up all possible strategies to solve your problem. And you have to be really consistent in scrutinising them all equally by similar, criteria. And so rarely that happens. So we will scrutinise GMOs, but we don't equally, that almost distracts attention from what insecticides might be doing. It distracts attention definitely from the fact that organic farmers can use pesticides, which we know a whole lot less about, and in some cases are far more toxic than the ones that we're concerned about that are synthetic. And it just seems to me that we need to be really rational about evaluating what is best in a given context, looking at issues like environmental damage, human health, economics, and just look for the best solution without having biases about what that might be.
So I wanted to take it beyond the human germline editing, because this conversation about regulation hasn't been talked about much in the Australian population, and so I think it's a really vital one. For example, one of the major reviews underway, there's still open for submissions until Thursday this week. So it's a very active conversation right now.
Go home and start writing.
Yeah.
So bringing it back though to human germline editing, because this is where things get very speculative but very interesting, and obviously it's dominated the public conversation because it tests our imagination, doesn't it? These questions of whether we'd use CRISPR for enhancement, for example, or designer babies, etc. Just to think about though a possible future where people could use CRISPR to modify their embryo and carry it to term and create a baby with the genetic traits that they desire. What about the prospect of genetic haves and have-nots? And who would decide who has access to this technology? And who would decide about what gets deleted and what doesn't?
Yeah.
Should it just be the parent?
Well, yeah, I think that's a really interesting question. And, we're talking about looking at each application on its own merits, and I would really like to see each particular gene modification you're trying to do looked at its own merits kind of thing. I think there are going to be cases, like I mentioned in my talk, about height or about cognitive traits where there would be this huge risk if you let everyone do it sort of, willy-nilly. But I think there are going to be particular genes where you do have good reasons to change, and to some extent, we should be able to let parents maybe have some sort of options within that sort of range. But,
it's tricky though parents are sort of, they're affected by the trends and desires of the time. Yeah. They're affected by social pressures. Many would argue that, for example, genetic diversity, including disability, is fundamental to cultivating a kind of diverse and compassionate society.
I completely agree. I mean, I think genetic diversity is one of the things we should try and be maintaining in these things. I think there's a real risk. Well, it's a complicated thing because not all diversity is equal. I think you want diversity in some traits, but not others. So, for instance, any diversity in sort of the genes that control how your heart pumps blood around the thing is really risky. If you're going to get diversity in those genes, you're likely going to have a bad health and die. But if you're talking about things like different cognitive traits, I think diversity is really important. And if you're talking about immune traits, diversity is really important. So it's about looking really at the details and about the particular application at that particular time.
Louise, your thoughts about the question of the haves and have-nots, because that's a social justice issue then.
Yeah, I mean, I think no one said the eugenics word yet, which I will, and I really think this kind of thing, especially if you base it on a trait-by-trait this decision, it really is a slippery slope to eugenics. I mean, I think I think, yeah, as you would have seen from the reaction of scientists, prominent editorials in Nature all called for a moratorium on human germline editing, and I think that's, the sensible approach given the current state of the science, given that there are off-target effects associated with these techniques. We're just so far from it being, them actually being able to do it. I mean, if you actually look at gene therapy, it's been being used for nearly 40 years now, and there's still not any, I mean, it's a really promising technology, but it's not been hugely successful because we keep coming up against these unexpected effects. I mean, there was a whole bunch of, there was a technique that looked really promising, and it was in use in trials in children, and then a whole bunch of the children got leukemia and they had to discontinue it. So I really think that
it's delicate terrain.
Scientists actually need to be aware of the limitations, and I think there's a lot of hubris associated with CRISPR and scientists thinking, oh yeah, it's great, we just do these tweaks, and it's much more messy than that. And when you're talking about human germline editing, it's irreversible, and it's also transferable to the next generation. So, I mean, these are people's lives you're messing with it. I find it really quite incredible that, I mean, the example you were using is, what if someone's got a genetic defect that they don't want to pass on to their children? Well, you can achieve that same thing through genetic screening. It would be a very, very tiny percentage of cases where someone might be homozygous for a particular trait. And in that case, what's so wrong with actually using a donated sperm, a donated egg? Is it really preferable to have a genetically modified baby than using someone else's egg or sperm to produce a child? It seems really quite bizarre to me.
I think there's really two
and then I'll come to you.
Sorry, there's really two things running here together, and I think it can really blur the discussion. So one is, yes, we want to make sure it's safe and there's no off-targets. That's an empirical question. That's a scientific question. That's not an ethical question. So the question is, if you can do it perfectly, do you have moral reasons not to do it? And I think it's a really strange idea to say, yes, it's completely fine to create a range of embryos, test each one, implant the one that you want, and that's completely fine, rather than just creating one embryo, correcting the defect, and that's all of a sudden, no, we've gone too far, we're eugenics. It seems morally really hard to draw the distinction there once you take away all those empirical concerns that are, of course, will be dealt by safety trials and through research.
So there's off-target effects, but there's also epigenetic effects as well. So there's a whole range of things that we actually don't understand about the genome, and we don't actually know what the implications of doing that until we bring live living human beings into the world.
Live human beings are coming into the world with their genes,
and
with their different epigenes.
Wheeling in the geneticist.
Yeah, so a couple of things.
Phil Batterham.
A couple of things here are really important. First of all, an on-target effect can be an off-target effect. So in the terms of that super mouse, there are a number of different traits impacted. To say that there's a gene that's for height and that's all it does, or for cognitive ability and that's all it does, that is going to be a rare circumstance. There is this thing called pleiotropy, and most genes that if you mutate them, then it has multiple effects. Secondly, for traits such as height or cognitive ability, there'll be hundreds to thousands of genes involved.
Multigenic.
So the notion of us doing anything about these things anytime soon, like in the foreseeable future, is just minimal. I just find this a very difficult discussion to have. It is good that we have the discussion in advance of us getting to that point in the future. But to think that it's anytime soon, to think that it would be simple is, well, it's beyond Gattaca and then some. It's so far out there.
So I'd like to open it out to the floor now.
This relates to regulation in general. In the last 30 years, we've been under the neoclassical or neoliberal model of regulation, which means remove regulations. It gets in the way of the market. Now, we've seen the consequences of that attitude in the recent global financial crisis that almost destroyed the world's financial system. And also recently the current drama that's going through our financial system here in Australia. So I guess it's really important that we look at the value of regulation for avoiding some of these consequences which can spill over into our biological activities as well, because if people are given free rein, then what control do we have over the consequences?
Okay, so more of a comment than a question?
Yes.
Great. I don't mind having comments. I think this is a good opportunity for us to have a chat, really. But if you can keep your questions fairly concise, thanks.
A couple of practical questions. Is it reversible?
Yes.
In principle, you can reverse it. Okay, I
Yes.
didn't know that.
And
you can
I would
insert.
question that, because I mean you can try and reverse it, but what if you then create additional mutations when you try and reverse it? That's
Well, I guess that just the principle, though. You can insert and you can delete, and in principle, it is reversible. Really interesting. Thank you for that. A comment. I think it's great that we've got opposing views here, or slightly different views here. I think it's extremely important. So my question is, we are polarised across so many issues around science, from GMOs, vaccines, etc., etc. How do we prevent this becoming the same kind of knee-jerk, GMOs are bad, vaccines are bad, global warming isn't, from those sections that don't listen to the science? How do we keep this conversation going so we don't fall into that trap?
Thanks. It's a vital question, isn't it? How do you have a mature public discourse that's respectful of all views, right to the very tippy tail of a regulation process as well?
Yeah, I actually think, you want an engaged, deep conversation, and I actually think if you keep these conversations going, eventually you get that. I have a feeling that me and Louise, if we had 6 hours together, we would come on get through our issues and get on the same page, because I think a lot of these differences, they come from different assumptions, they come from, different background thoughts that you can get clear about.
But they also come from places where there are conflicts of interest too, and that is an issue in gene regulation around the world. There's no doubt about that. You can't deny it. People are on these committees with an interest in the technology.
I can only think of one example where we've done well in scientific debate and reaching some consensus, and it was around stem cell legislation. And you had a situation where politicians became really embroiled in this issue. You had a situation where Prime Minister John Howard changed his position. You had state premiers speaking up, and at the end of the day, a conscience vote in Parliament voted in favour in the same proportion, basically, as the Australian public were in favour, as measured by Gallup polls. But I can't think of any other situation where we've had that kind of national dialogue that's run all the way through the community, through to Parliament, and ended up with, a consensus decision made that we seem to be happy to live with.
Yeah, the government actually had an interesting process through the National Enabling Technologies Strategy. So they looked at, they did a bit of work on synthetic biology, and they did a bit of work on nanotechnology as well. And that actually ran a series of sort of roundtable discussions. Members of the public were invited along, a series of experts kind of presented, and then the public were basically left to say what was important to them. And I thought the findings were really interesting. There's this assumption that the public don't have clear views on this, that they're uneducated, but actually,
or that they're not capable of synthesising complex information, which I would argue that they are.
Which is deeply unfair, I think. And they actually, they came up. They just said that they wanted nanotechnology to be regulated, they want it to be labelled. There's a whole range of other recommendations, but the problem with that process is it was completely disconnected from the rest of government, so it wasn't feeding into the decision making.
Actual policy and legislation.
And I think that, but I actually thought the process itself was good. There just wasn't buy-in from the Department of Industry.
And there's really great processes in Europe called consensus conferences, which we've trialled once here on GE, and it didn't really ever go anywhere, that process. Lots of hands are up. Sorry, go. Yep.
Chris mentioned Josiah Zayner in his conversation, the biohacker in the USA who injected himself, and a number of other biohackers have done the same. Now, in Australia, there are biohackers as well, particularly BioFoundry, which is a so-called citizen laboratory in Sydney, and there's a group here in Melbourne as well of biohackers. The OGTR and the National Review of the Gene Technology Scheme, which is going on at the moment, and as you said, closes on Thursday, does not appear to propose regulation of these people. In fact, in the discussion paper, they call themselves, they call the biohackers DIY biologists. They have no training, their facilities are very poor, and this is a case for regulation, I think.
Some of them do have training, but thank you.
Hello. I've spoken to farmers and doctors and citizens from North America, South America, India, the Philippines, around how genetic engineering in agriculture has affected their lives. And it is incredibly terrible. And if you see what's happening in Argentina or in Sri Lanka, you've got people dying from poisoning from glyphosate. You have the chemicals everywhere. This is the industrial agriculture. I've just got back from the Philippines, and what you find is that the farmers there do not want any of these genetic engineering. They do not want the genetically engineered rice.
That's a separate issue from glyphosate.
Well, no, but what I'm saying is that what you're talking about is genetic engineering in agriculture and saying this has all been fantastic and people are fear-mongering about it. What I'm saying is that I've actually heard from the people in Sri Lanka, in South America, in North America, and what the genetic engineering has brought them so far in crops is increased pesticide use and increased pests and a huge amount of problems and illnesses. So this is tens of thousands of people and it's documented.
Okay.
Therefore, I think that before we run down this route, we need to have a very careful look at what has actually gone on worldwide.
Okay. Thank you so much. So would anyone like to comment on...
Yeah.
Well, I would comment on that last comment. I would go back to what I've said and what you've said, that each technology and each application of it you need to evaluate on a case-by-case basis. And you can't say that a technology is no use across the board because there is one application of it that you do not like, or that others do not like, or has proved to be ineffective, or so on. So I think that's just really important. In terms of people going feral and using this technology on themselves, that can't be stopped by regulation. Those are people who are clearly just moving outside the bounds of common sense and regulation. So there's been a long history of people doing those things. Sometimes it's been led to great advances in medicine.
Sometimes they're community art projects. I mean, it's not...
absolutely.
It's not quite as... It's not quite as cowboy as you're making it out to be.
What could happen, though, what could happen is in this area of gene drive, where basically you're sending a CRISPR mechanism of control viral, and that word is maybe a little too inflammatory, but it spreads. If you have an insect pest that has large range, then if someone did it in Southeast Asia, it would impact us. And our regulations here would not control it. And that is something where, you know, their perspective on the problem might be different. You know, it might be in a developing nation where the problem for them is enormous, has devastating impact on their agriculture, and they would view it very differently than we would in our nice, wealthy Australia. So we've got to realise that regulation has borders, and sometimes these technologies don't.
Yeah, I'd just like to also say that, you know, it really touches on the sort of philosophical basis for regulation here. So I think if you can think about some of the biohackers, if they're just getting the thing themselves, they're injecting themselves, and there's no harm to others or no possible harm to others, there is the argument they should be free to do that. Normally we think states are only justified to intervene if there's a risk of harm to others. But for a lot of these technologies...
How do you assess that harm?
Well, that's the
the
interesting
trick.
question, yeah.
I'm going to take 3 questions in a row, and then we probably need to wrap, I think. So I'm just going to whip through 3 questions, maybe 4, but in a row. So, and we'll try and remember them. So pick a question to respond to, I guess, might be the go. Thank you.
Hi, thanks for
So keep them very tight so we can move through the crowd.
Gotcha. I'm
Thanks.
sorry.
No high.
Yeah, I guess the discussion so far has been about regulation in Australia, but there's no body that regulates the whole world. So what's sort of the way forward with regulating internationally?
Yeah, good question. Next question.
Hi. Why do you think that there needs to strictly be a specific or unique regulatory framework here? Technically, existing avenues to legal recourse are able to capture the harms that arise here. Things like torts, negligence, consumer law may be able to, or will be able to capture these kind of harms. Why do you think there needs to be a specific regulatory framework? And as an extension of that, is there a risk that it might reinforce public concern that there is some kind of unique risk pertaining to this technology?
Interesting question. One more here. I've got it.
So just in relation to the regulation, do you have any sort of practical sense as how to do that? I manage an ethics committee and a biosafety committee. We're really under-resourced, underfunded. I'm terrified of having to review these applications. So how are we going to practically fund this regulation?
What a vital question. Is there one more question? Or, yep, I've got it.
Okay. Where's the Wild West of this technology at the moment, and what's happening there, and why should we be worried?
The Wild West. All right, so there are 4 comments. The first one was on global regulation, given that we're crossing multiple jurisdictions. How do we approach that challenge? Do we work collaboratively?
Yeah, I think that's a really key issue, and I'm thinking more now about the germline gene editing aspect. So the National Academy of Science did their review of the ethics of gene editing, and especially germline gene editing and somatic, and they identified that as a crucial problem, and with no solutions that they could think. The only thing that you could think to do is to try and get like a UN-bodied charter that people sign up to, but obviously that's going to be really, really hard. And I guess it relates back to the Wild West question too. In China right now, you've got a lot of gene editing going on, not just in embryos, but you've got somatic trials. It's a really different regulatory system there, and you know, there's nothing you can kind of do for these sort of large concerns about the human gene pool if you don't have a human gene pool regulator as it is. So I don't know the answer.
Any other comments on, we've got the Wild West, we've got, is specific legislation really needed at all for this technology? Would that actually put people off the prospects of this technology? Our ethics committee chair, that's a vital question. If they're not being resourced to review these adequately, what happens? Any thoughts?
Yeah, I'll go for all 3, 4. So at the international level, there's actually UN discussions going on in the Convention for Biological Diversity on synthetic biology, which includes these new techniques, and also techniques such as gene drive. So there's actually been calls from 160 global organizations for a moratorium on gene drives, and that's been supported by a number of developing countries at the UN. So there's actually the next round of discussions, I think, is in June, so it's coming up soon. And interestingly, Australia has actually been playing a blocking role along with the US and Argentina and a few other pro-GM countries. We're actually with that block. Our government is actually trying to block any discussion and any moratoriums on gene drives, which is interesting. As far as the Wild West, I would argue that the research that CSIRO and University of Adelaide are wanting to do using gene drive mice in the Wild West is actually, we find that really deeply concerning. And also the fact that one of the members of that project actually is one of 3 academics advising the federal government on how these techniques should be regulated, or even whether they should be regulated at all.
I'll pick up on the questions of regulation. I believe regulation is necessary. I believe it's important for scientists to conform. And in terms of it being funded, basically you have federal regulations, and then organisations like the university I work with really has to find the resources to impose that level of regulation.
So what about the under-resourced ethics committee? They're the coalface.
Ethics is different, so I've got no expertise there, but I know that our research is scrutinised. So when I say that I'm in favour of regulation, it comes at cost. There is the pile of paperwork, there is the inspections, there is compliance, but I still believe in it.
Well, look, we're going to wrap the night. I'd like you to thank our panellists, Louise Sales, Phil Batterham and Chris Gyngell. Thank you so much. And I'd also like to say that the Convergence Science Network extends their great thanks and appreciation to the sponsors of these events. They take a lot of work to put on, and we are particularly grateful for you coming out and being part of them and part of this rich discussion about such vital issues. Thank you. Have a great night.